Okay guys, so for the last little bit of class, what I want to do is return to the case study that I asked you guys to read for homework, at least the first part of the case study. There are 3 parts of the case study. And you guys answered 5 questions, you turned it in. I'll be sure to grade those for you and pass it back to you next class period. But there was one question that I asked you as you were coming into class today in your groups of 4 to put up on the board, your list, that was tackling a particular question. And before we get to that, let me just remind you guys that that first part of the case study was focusing on chimpanzees and kind of the society of chimpanzees. You know, we learned that they're omnivores. They don't just eat vegetation, berries and leaves. They can also hunt in groups and hunt monkeys, for example. We were also reminded that chimpanzees are a male-dominated society and that oftentimes we have groups sort of dispersing in the day, but at nighttime they always come back together where they're in close quarters with each other. And so what we're going to do is sort of build on that for the rest of the class period and sort of think about how chimpanzees might give us some insight into evolution. So let me just sort of dim this so that we can take a look at that one question in particular that I had you write your answers to. It says, Dr. Hahn and her associates are specifically collecting fecal pellets, and you'll recall this is in 3 countries in Africa. Why do you think they might be interested in the contents of the fecal pellets? And I can see from this list that you guys put up over here, it looks like there's probably about 3 different categories of information. So we have the idea that you might be interested in finding out what they're eating, right? So we can look and see if there are seeds, if there are particular leaves that didn't get digested, if there are bones in there. It also looks like related to that would be nutrients. So is there a particular nutrient that's found in high quantity in one of these food items? Or maybe you can calculate calories and get an idea of how many calories they're taking in per day. And then the third one, that it's not as common up there, but I'm glad that some of you thought about the idea of parasites, because it turns out that this is what Dr. Hahn is interested in. And what we're going to do is think specifically about viruses that might be found in fecal pellets. Now, it turns out that Dr. Hahn is interested in HIV and the evolution of HIV. And this is a type of retrovirus. And retro, if you think about that term, retro, of course, means kind of to step back, right? So we use that term when we're thinking about the 80s, and we're stepping back to the 80s. But when we're thinking about it from a virus point of view, we have to kind of backtrack into some information. And this is going to become more clear as you read through part 2 of the case study. But before we get to that part 2 of the case study, I want to show you a video clip. It's a short video clip. It's about 3 minutes long. And as we watch this video clip, again, I want you to think about what is the virus doing that allows it to take over the host cell.
Comes the virus, and it's going to land on one of this guy's throat cells. So notice it's covered with little yellow knobby things. You call these keys, right?
Those are the keys.
Yeah. This is a key, this is a key, this is a key.
Okay, if the keys on this virus happen to fit the locks, which are those little purpley stick-uppy things on the surface of the cell, if there is a match, the cell, watch this, welcomes the virus in. And what's this?
This is the welcoming committee. They all interlock with each other, and they pull this membrane down into the cell.
And down it goes deeper, and that welcoming structure disperses, and the virus capsule bursts, and out comes the secret recipe for how to make more viruses, those little noodly things. So this unsuspecting cell has been tricked into guiding these virus recipes right into its own command center, the nucleus.
Okay, so as we saw from that 3-minute clip, there were 3 things that are quite significant for how a virus is going to successfully take over a host cell. And this is a really simple diagram of a pretty complex process. But because we're in intro biology, we're just going to keep it at that introductory level. And as you guys go on and take your more advanced classes, you'll get some more of that information. But there are 3 things that I want you guys to be sure that you have written down in your notebooks. The first thing, as the narrator indicated, is that the virus has to be able to fool the host. It has to enter the host, it has to bypass the immune system and get into the host cell. Okay? So the first thing, the virus has to get into the cell. The second thing that happens is that the virus has to get its DNA into the host DNA. And then the third thing is that the virus then, now that its DNA is inserted, the host can start to make more virus particles rather than host particles. Okay, so again, the first thing, the virus has to enter the cell. The second thing, the virus has to release its DNA into the DNA of the host cell. And the third thing is that the host will now start making virus particles because it has that DNA in it. Okay, so what we're going to do next is we're going to tackle the second part of the case study, which is going to sort of take a look at HIV and sort of think again, how is retro involved as you're working through this case study? So I'm going to ask you guys to get back in your groups of 4, and as you're reading through the case study, which is just several paragraphs long, you're going to be generating a list, okay? And let me put this back up on the board here so you can see what I want you to be doing. You and your group members are going to generate a list comparing and contrasting RNA and DNA. And then I want you to answer the first 4 questions of the case study. Okay, as always, I'll ask you guys to nominate a secretary to record your answers, and then you'll be turning that in as a group at the end of class, and I'll grade it and give it back to you next class period. Okay, so go ahead and get yourselves into your groups. Start working on that. I'm going to walk around, kind of look over your shoulders, and help you as needed. Okay, so now that we're back after your group work, here is the list that you guys generated, thinking about RNA and DNA. So RNA and DNA are both made up of nucleotides, which have these components, phosphate, sugar, and base. And it turns out that the base is really important, right? We've got adenine, cytosine, and guanine common to both DNA and RNA, but thymine is unique to DNA, uracil is unique to RNA, and it's those bases that become really important for making these proteins, right? So DNA provides the information, but the RNA helper comes along and helps ultimately convey that into the protein. That's really, really significant. Other differences that you guys noted is that we have a different sugar in our DNA and RNA, and the names of these molecules, of course, are based upon that sugar. Deoxyribose for DNA, ribose for RNA. And then the last thing that you guys said is that RNA is only 1 strand, DNA, of course, is a double strand. And when we go back to thinking about HIV as a retrovirus, what's significant is that HIV is RNA-based. And so HIV has got to go back to make a copy of DNA, because remember, the DNA is what gets inserted into the host cell. And so again, we're going to come back to that as we work through part 3 of the case study. And because we're running out of time today, what I'm going to ask you guys to do for homework, I'm going to pass out part 3 of the case study. And for homework, what you're going to need to do is read through that and come prepared to deal with the questions in class next time. However, I want you to specifically go to this site. I'm going to post these slides on our class site. You can click on this link, and it's just a few minutes long. I'm going to show you what it looks like. And when you go here, you just press play for the animation, and then you'll notice this tab that says quiz. There's about 3 questions for you to answer. And just sort of sit with it. We've already talked about evolution in this class, but you know, it's one of these things that people think they get, and then once they start trying to explain it to somebody, they realize, maybe I don't understand it as well as I thought. So with that in mind, what we're going to do first thing before we start talking about part 3 of the case study is I'm going to have you guys answer, stand up. I'm going to randomly pick different groups to address these questions. What is a selective pressure? What's the difference between adaptation and fitness? And so I'm going to have you guys being the teacher and explaining these concepts to your peers. And for those groups who really impress me, who can do a really good job explaining this, I'm more than happy to give you guys a little extra credit points. Okay, and then finally, before we break up, I just want to remind you that, as always, at the end of these modules that I post, there are study questions for you to consider. So even though we haven't gotten through part 3 of the case study, like this particular question deals with part 3, there will be questions for you to make sure that you understand the material, and if you don't, that you can get help from me before the next test. Okay? So I will see you guys next time.